All right, everybody, welcome to the last lecture of the quarter. Today's topic is going to be translation. And this topic is going to bring us full circle. We've completed our talk about biological information and nucleic acids, and we're going to try to tie it back in with the beginning of the quarter when we were talking about protein synthesis. So what is translation? Translation is the synthesis of a polypeptide, that is a protein, based on the sequence specified by an mRNA nucleotide sequence. So you'll recall that in the last lecture, we talked about transcription, which is the process of making an mRNA copy of the coding strand of DNA. And we said that transcription ends up giving us an mRNA molecule which has a certain sequence of nucleotides. So, for example, A, G, U, A, C, 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 G, U, and so forth. We said that each three-letter combination is what's called a codon, and a codon specifies an amino acid in a protein to be made. But in order to make the proteins, we need to, first of all, find some way to get our amino acids all together in one place, and then we need to somehow translate the codon code to an amino acid code. And the key to do that is with tRNAs, which become linked to amino acids matching the anticodon of the tRNA. So the example we gave last time for the codon was AGG. So this is what would be on the mRNA. And we said that the anticodon would be CCU. And that's what we have on tRNA. And our tRNA molecule is a big, long, complicated molecule, but at the top of it, we would have the amino acid attached, which matches the anticodon of CCU. And we said that's arginine. So today we're going to take a look at how you actually use this anticodon-codon pairing to make proteins. So translation is protein synthesis based on the sequence encoded in mRNA. This is done using the ribosome. The ribosome is your catalyst. So the three things that you need for protein synthesis. First of all, you need an mRNA. That specifies the sequence that you're going to be putting your amino acids in. You need an amino acyl tRNA, or a set of amino acyl tRNAs. Those are your raw materials. And the last thing that you need is the ribosome, which is a catalyst, an enzyme, if you will. So as we did with replication and transcription, we're only going to talk about translation in bacteria. So the bacterial ribosome is a two-strand RNA with several proteins bound to it. So we say there's two subunits to the bacterial ribosome. We have the small, which is also called the 30S, and we have the large subunit, which is called the 50S. This diagram shows a side view of the bacterial ribosome. The 30S subunit is in tan and the 50S subunit is in aqua. If you rotate it, you get this diagram that we see here, and you'll notice that there's a hole running down the middle. As we're going to see, the mRNA passes through that channel in the middle of our ribosome. And what ends up happening is that the ribosome moves along the mRNA sort of like it's reading tape through a tape machine, and the protein gets spit out the side. And the protein, as it is being synthesized, threads its way through this tunnel that's shown at the bottom right. We'll talk about that in a moment. So the functions of the ribosome are many. So it's not just a catalyst, it's also an assembly platform for all of these materials that we need. First of all, we have to pair up the tRNA with the mRNA so that we've lined up all our amino acids in the correct sequence. You have to get your ducks in a row, in this case quite literally, except not ducks amino acids. And then secondly, you have to actually form peptide bonds between the amino acids. So those are the processes that occur at the ribosome, and we're going to look at them in detail. So before we do that, let's take a look at the geometry and the layout of the ribosome. So once again, we have the small subunit on the left and the large subunit on the right. So the ribosome has three important binding sites. We'll say binding and active sites because there are catalytic steps that occur at each of these three sites. A, P, and E. So they're easy to remember because they spell ape, and they end up getting used in that order. So the A site, I remember this as calling it the access site. And that's what's shown in yellow, or excuse me, that's what's shown in green. This is where the amino acyl tRNA site initially binds, with one exception, which we'll see in a minute. So that is where the amino acyl tRNA binds to the mRNA corresponding to its anticodon. Moving into the middle of the ribosome, you have a second site called P. P stands for peptide bond formation. And that's where the amino acid from the tRNA gets added to the growing polypeptide chain. E is the last site, that stands for exit, and that's where the empty tRNA, which has lost its amino acid because we just added it to the growing peptide chain, exits. And they're laid out in order in the diagram that we've shown at in the middle of this page. So to summarize, the A site pairs up the AAtRNA with the mRNA. The P site is where the amino acid gets transferred from the amino acyl tRNA onto the growing polypeptide. And the E site is where the empty tRNA gets released. All right, how does the tRNA pair up with the mRNA? We always said before it is base pairing between the codon for amino acid X, that's on the mRNA, and the anticodon on the loop of the tRNA for X. So in this diagram, if this is our mRNA, and this is tRNA for amino acid 1, it's going to have amino acid 1 connected to its 3' end, because that's how it works. So if our mRNA codon is, say, GCC, then the anticodon would be GGC, right? We're reading from 5' to 3'. Okay, so this is the 5' end of our mRNA, of our tRNA, excuse me, and that's the 3' end. So on this mRNA, we're actually showing it backwards. All right, just for convenience. So what's going to happen is this amino acid for tRNA1 will base pair briefly with codon 1. And then at the same time, the tRNA for codon 2 will base pair with the codon at codon site 2. And what happens next is the amino acid 1 gets transferred onto amino acid 2, so that we end up building up our polypeptide chain from N-terminus to C-terminus. This is possible because you'll remember the amino acids get connected to the tRNA through their C-terminus. So when amino acid 1 gets transferred onto amino acid 2, we end up forming a peptide bond and amino acid 2 now is attached to tRNA. And there's our N-terminus. So an important thing to remember during translation is that a growing polypeptide is always attached to a tRNA. So the function of the mRNA is it gives us a sequence of codons. Our tRNAs just line up in rows on codons, and then the ribosome transfers each successive amino acid to the next amino acid. All right, let's take a look at the initiation of translation. You are responsible for knowing the general features of translation initiation, because translation initiation is, important. How do we know that we're starting our protein at the right spot on our mRNA? We have to be sure that we start at the first amino acid in the protein and don't just start at some random location on the mRNA, because then we're going to make a protein that's too short. So this is analogous to the problem of starting at the promoter in transcription. So how do we initiate translation? Well, the way it works is fairly complicated. So I'm going to give you guys the short version. It starts with the small subunit. So the subunit binds two proteins that are called IF1 and IF3, and they bind at the A site and the E site respectively. There's the A site, there's the E site. So IF1 binds at A, IF3 binds at E. Okay, so we've got these proteins here. What are they going to do? Well, what happens next is once these two proteins are bound, then the mRNA can bind. So we have IF1, IF3 bind to mRNA binds. And notice the way that the mRNA lines up with the sites on the ribosome. We have the 3' end hanging out loose in solution, and the binding of the ribosome occurs near the 5' end of our mRNA. And why does it do that? Well, it does that because the 30S, the small ribosome subunit, contains this short segment called the 16S rRNA. It's one of the molecules that makes up the 30S subunit. You don't have to know what that is. But the importance of this 16S subunit is that it base pairs with this segment of the mRNA called the Shine-Dalgarno sequence. So what's that? So the Scheindel-Garneau sequence, shown in orange here, this is a present that's near the beginning of every gene in bacteria. And you can think of it as almost like a promoter for translation. It comes at the beginning of a gene, it base pairs with this 16S rRNA, and that allows the start codon to be positioned at the P site. So the function of this Scheindel-Garneau sequence is to base pair with the 16S rRNA so that the start codon, which is always AUG, we said last time that the start codon for the first amino acid is always going to be methionine, that's going to pair up at the P site. So the function of this is to make sure that our mRNA is oriented the correct way. All right, so once we have this mRNA bound with the start codon at the P site, what happens next? What happens next is this. We have a third initiation factor, IF2. It binds to the ribosome. And then once it is bound, the tRNA for methionine, the methionine tRNA, binds to the P site. It doesn't easily bind unless IF2 is present. Now, I'm skipping over some details here. It's actually a formal methionine, not plain methionine, but who cares? So the methionine tRNA binds at the P site. Once IF2 is bound, it base pairs with our start codon, and now we have everything in the correct place. All right, so once we have done that, what happens next? Well, we only have half of a ribosome. So what's going to happen in the fourth step is that the IFs dissociate, and this is a GTP-dependent process, so I'm giving you guys sort of the short version. And the large subunit binds. So what are we left with now? We have what's called an initiation complex. We've got our mRNA with the start codon at the P site. In the P site on the large subunit side, we have a methionine tRNA at the P site, and we have empty A and E sites. Now we're ready to begin translation. And so now we start translating. Okay, so this is the reaction cycle of translation. This is the most important thing for you to know about translation. But luckily, it's fairly simple and straightforward if you remember the initiation part. So what do we have? This is the complex at the top that we started with at the end of the last page. We have the methionine tRNA bound at the P site right after the IF proteins leave. And we've got empty A and E sites. So what's going to happen next? We have the amino acyl tRNA for the next amino acid. And what it's going to do is bind to the A site. Remember, A for access. And that gives us this structure that we have right here. So now we have amino acid 1 and amino acid 2 bound right next to one another. Okay, so we've got both amino acids that we need, the first two amino acids in our protein lined up side by side. So what happens at the next step? What happens at the next step is the tRNA that's at the P site transfers its amino acid to the A site tRNA. So notice the P site is going to be amino acid 1, the A site will be amino acid 2, and so what we're going to do is transfer amino acid 1 onto amino acid 2. And now we have a 2 amino acid peptide bound to the tRNA that's at the A site. The P site now has an empty tRNA in it. Next step, the mRNA sort of clicks down one level, feeding through on the ribosome, just as I said, sort of like tape feeding through a tape machine. So that means that our empty tRNA now moves over to the E site, and when it's in the E site, it's kind of open to the surrounding solution and it doesn't stay there very long. So it exits. What are we left with now? Now we have a 2 amino acid peptide bound to the tRNA that's now at the P site. So this kind of looks like the complex that we started with, where we've got an amino acyl tRNA at our P site, but empty A and E sites. So all we have to do now is repeat the process. The next amino acyl tRNA will bind at the A site, and then we're going to take our 2 amino acid peptide and transfer it onto that one, giving us a 3 amino acid peptide. The ribosome moves down by 1, the tRNA that was bound unbinds, and we start over again. Okay, that works great until we get to a stop codon, but we said last time that there's no tRNA for stop codons. So what do we do if we reach a stop codon? Well, stop codons, as you may have guessed, that signifies the end of the polypeptide. So we are done making the polypeptides and we don't have to make any more. So what are we going to do at that point? Well, since there is no tRNA for stop codons, we have to have some other way of making sure that transcription comes to an end. And so what ends up happening is that there are these proteins called release factors that bind to the codons in the A site. So release factors are proteins that bind to the mRNA similarly to a tRNA. And there are 2 release factors that we care about, RF1 and RF3. And these 2 bind together in a complex with GTP, sort of like the G proteins from epinephrine signaling. Right. So what happens is this. Release factors 1 and 3 in complex with GTP bind at the A site. So now we end up in this complex where we've got a protein at our A site, we've got our finished polypeptide trailing out from the P site. What's going to happen next? What happens next is the RFs add water to the end of the polypeptide chain, releasing it from the tRNA. So in other words, the RFs release the new protein, which floats away. Great. Now what? Well, after the RFs release that new protein, what we have is an mRNA, an empty tRNA, and these RF proteins all stuck together in this ribosome complex. And our ribosome complex is now not doing anything because it's already completed its job. So as it turns out, it's complicated to make a ribosome assemble, and it's also complicated to make it fall apart. So what happens next is that we get a GTP hydrolysis. And once we have hydrolysis of GTP by the RFs, a third protein called RRF, ribosomal release factor, is able to bind to it, and it simply breaks the whole complex apart. This is complicated. We're not going to get into details about how it works. RRF breaks the complex apart. So to summarize, RF1 and RF3 bind at the A site. RF1, RF3 add water to the aminoacyl tRNA at the P site, which releases the protein. And then thirdly, RRF breaks up the complex. All right, when I do this talk on translation during the face-to-face part of the class, I really like to do it by showing video animations as I go because I think that makes the whole process much clearer. Unfortunately, the translation videos that I've been able to find are not very good. So this first one is decent, but it doesn't show the termination step, which we would like to see. The second one is, I think, a little bit too, hey, look at all this cool stuff that we can do with this animation software that we just hired somebody to make videos with. The educational quality is limited. The third one is a pretty good balance of all of them. I think it's maybe a little bit oversimplified, but I think it's pretty good. I'd rather oversimplify this topic than overdo the detail. It's worth mentioning that if you look at video 2, it's actually showing eukaryotic translation, which is a lot more complicated. So eukaryotic translation, as opposed to bacterial translation, involves a lot more proteins and different enzymes. Whereas in bacteria, everything is simple and economical. You just got your ribosomes and there's not much more on top of that. So what you are responsible for knowing as far as translation goes is know the steps of initiation, the steps of termination and ribosome release, and then the reaction cycle that occurs during translation, which is quite simple. And then I'm usually fond of hypothetical questions where you have to predict what would be the effect of some sort of defect in one step. And that goes for replication and transcription as well. There's some homework questions related to that. All right, you guys, we don't have any more material to go. This is the end of summer 2020, Chem 313. Those of you who stuck with me for the whole time, I congratulate you. You did a good job, and I know doing all this in 5 weeks is brutal, but you made it. Okay, so we've got our final exam that's going to be on this Friday. Looking forward to being able to give out plenty of A's, and it looks like people's scores are coming up on the quizzes, which is great. So stay with me. I will see you guys in office hours, and be looking for a final exam study guide.